All right, today's show cuts about as close to home for me as it possibly could, and probably does for you, too, because Alzheimer's disease is just so prevalent. It's just very unlikely with 5 million or so Alzheimer's patients in the United States that you don't know somebody, that you haven't followed somebody's story. It might be somebody in your life. You might have also been a big fan of Tom Magliozzi from Car Talk here on this radio station, or watched the career of Pat Summitt. For me, the story kind of started earlier in this decade when my mother began to forget things. I took her to a neurologist, and he said to her, "I'm going to say 3 words to you, and I want you to remember them because I'll ask you about them in just a little while. Banana, chair, sunset." And then he asked her a whole bunch of other things, and in the most charming manner possible, she revealed how little she could remember. And she'd kind of been, I don't know, she'd managed to disguise this from me. I was very closely involved with her all the time, but I just wasn't, somehow or other, she was very good at hiding it. And it was really breathtaking. She came from a little town in Massachusetts, which was put underwater, part of the Quabbin Reservoir, and it seemed like her whole life was kind of underwater as she tried to answer these questions. And then the doctor said, "Do you remember any of those 3 words?" And she said, "What 3 words?" And that was the beginning of our Alzheimer's disease journey. And banana, chair, sunset kind of became my mantra for our Alzheimer's disease. But everybody has their own. And that's not the reason we're doing this show. And we could have lots of reasons for doing this show because, in fact, there's so much interesting research being done on Alzheimer's disease that almost, it seems anyway, as though every week produces something. And there's within this week, there was this enormous dump of research on epigenomics that had some pretty significant implications for Alzheimer's disease. But the real reason we're doing this show is because I got an email from one of the science teachers at the place I went to high school. And his wife has Alzheimer's disease, and he, being a science teacher, was really interested in the modern science of it. You know, what's going on right now? He's really been following this stuff. He may call in at some point during the course of the show. And I hadn't really thought about it that much. My mother died in 2006, and I'm not a scientist. I mean, I wish I'd been a good scientist. I wish I'd learned all the things that he could have taught me, or when I went to Yale, I wish I'd learned some of the things that our guests today could have taught me. But I didn't. But the science of this is really interesting. And fortunately, we do have a producer here, Betsy Kaplan, a nurse who can understand the science. So she's helped to put together a show today. With us in studio are Stephen Strittmatter. He is professor of neurology and professor of neurobiology and director of the Memory Disorders Clinic at Yale. And even though I don't have a memory disorder, I can't remember the other title you just told me that was really hard. What was that one?
Director of Cellular Neuroscience, Neurodegeneration, and Repair, a laboratory.
Yeah. If you were director of Banana Chair Sunset, I would have done a whole lot better. But and with us also Christopher Van Dyke, a professor of psychiatry, neurology, and neurobiology, and director of the Alzheimer's Disease Research Unit at Yale University. So, Christopher Van Dyke, I'll start with you. What is Alzheimer's disease? I mean, is there now a comfortable thumbnail description? I know we're going to set early onset Alzheimer's aside for a second and kind of talk about the animal that hits us from 60 years onward. Do we know what that animal is now?
Well, we know a lot, and we know a lot more than we used to, and there's a lot we need to know. So I'll start with just a little bit of the description, what it's like in people, and probably Steve can add in more about some of the pathogenesis. But so Alzheimer's is the most common cause by far of dementia in older people. And maybe we should say that what dementia is a loss of cognitive abilities that's bad enough to affect a person's daily functioning. So Alzheimer's starts, again, typically between the ages of 65 and 85. It can begin earlier. Most common early symptoms, Colin, are just what you describe. It's typically a loss of short-term memory. Person becomes forgetful. They may start to repeat themselves in conversation because they can't remember what they've said. They may become disoriented to date. So, fairly innocent short-term memory symptoms that get steadily worse. They go on to involve higher cognitive abilities like language, communication, visual spatial ability, start to impact a person's daily living skills, usually first things like driving a car or doing finances, things that are more difficult, and eventually go on to involve basic things like cooking, chores, dressing themselves. Probably about a 10-year course on average, although it varies a great deal.
Just to interrupt for a second, my experience there with my mother was that a neurologist examined her, and he didn't say she had dementia. He said she had Alzheimer's disease. There's other kinds of dementia. There's vascular, there's Lewy body, there's all kinds of stuff. So not having any lab panels or anything, I mean, how did he know she had Alzheimer's disease?
Well, he didn't. He didn't, for sure. But and one of the things we can talk about, in terms of specialty clinics, such as the one that Steve is directing, we do an initial workup. We take a history, which is so important. Then we do cognitive testing, mental status testing. We may refer the person for very in-depth cognitive testing with a neuropsychologist to really delineate just what they can and can't do. We do a bunch of laboratory tests for so-called reversible causes or other medical causes of cognitive impairment. Do specialized brain imaging. Things like MRI, structural imaging may involve even more specialized imaging. And then there are a range of more specialized tests. But actually, why don't we let Steve jump in here on some of the things that they do? So.
I was going to go to Steve next and say, but let's go. Okay, so Chris started us on the outside, what we see in the patient. But pretend for a moment that you're talking to a reasonably gifted middle school science student, which is probably about where I am right now, and maybe a lot of our listeners too. And as best you can, then sort of sketch it out for us. What's going on? What do we know that's going on inside the brain?
So when we see people clinically, we define their disease as probable Alzheimer's, but we say somebody has definite Alzheimer's when the brain is studied under a microscope, and usually this is after somebody's deceased. There's a particular appearance under the microscope which defines Alzheimer's disease, and that's the presence of amyloid plaques and neurofibrillary tangles in a certain density, and this makes the disease, the diagnosis. This is something that's been known for 100 years. We know that these things are present. They define the disease pathologically. Where all the research is going is to understand how those things we see in a microscope affect the biochemistry, the synapse function, why they manifest themselves in the symptoms that we see amongst our patients.
I just want to roll back the tape for a second. One of the phrases that I encountered a lot doing the reading for this was amyloid plaques. But once again, at the level of a 12-year-old, reasonably gifted middle school student, what is an amyloid plaque?
Right. So they're not the normal appearing brain. The brain is full of neurons, supporting cells, synapses. In Alzheimer's disease, there's huge blobs of protein. You can think of them as goo. They accumulate. They're bigger than a cell. They're huge balls of protein, and they're composed largely of one molecule chemically, and this is the beta amyloid peptide. That's why they're called amyloid plaques. So they can fill up as much as 2 to 5% of the brain, and then they cause the neurons all around them to stop working normally.
We'll come back to that in just a second. But first of all, I want to say if you have questions or comments, 860-275-7266, 860-275-7266. You may also tweet us at WNPR Colin. And Christopher Van Dyke, somebody has tweeted us with a really great question. Nancy tweets, how important is it to get a diagnosis of Alzheimer's versus other kinds of dementia? So, as we alluded to this, there are other things that present as dementia. How important is it to know that your relatives or your own dementia is Alzheimer's disease?
Well, I think it's very important, and I think it's going to become even more important when we get very effective and specific treatments for each of the dementias. But right now, the drugs that we have that are approved, that are fairly modest, they're approved for Alzheimer's disease. They're not approved for frontal lobe, frontotemporal dementia. They do have some benefit in vascular dementia. Many people think they definitely have benefit in Lewy body dementia. Most experts think, but it can help in that way to know how to treat. It can certainly help to know about the prognosis and the other associated symptoms. I think it will become highly important when we get, let's say, we have a highly effective anti-amyloid treatment, referring to the protein that Steve's discussing. That's likely to be effective in Alzheimer's disease. It's likely not to be effective in frontotemporal dementia. So going forward, it's going to become extremely important to know the difference.
So, Stephen Strittmatter, how the $64 billion question is, what's the pathway, right? How does this stuff get in somebody's brain? How does this stuff that we don't want to have? See, this is how I think. This is the level at which I have to express this stuff. How does this stuff that I don't want to have in my brain get in my brain? And one of the part of your research is to fill in gaps in the understanding of what that pathway is.
That's right. Let me just clarify one thing before I answer your question.
Sure.
Is Chris's answer is absolutely correct for the vast majority of people, but one of the things that's critical and to get out to your audience is there's a small number of people who have treatable causes of dementia. Mm-hmm. They may, if it's confirmed they have Alzheimer's, they're in the boat that we were just talking about, but there's a small subset, maybe it's only a couple percent of people who have treatable causes of dementia, and their whole life may change if that turns out to be the case, and they don't have Alzheimer's disease. Okay, back to the question about amyloid. You're right. My research focuses largely on how amyloid damages the neurons in the brain. But you asked the question, how does it get there? Why does it pile up? What's going on? And the short answer is we really, unfortunately, don't know that in detail. We know it for a small number of patients who have mutations in genes that are involved in the amyloid process. That's probably only 1 out of 200 people with Alzheimer's. It's the people who have early onset Alzheimer's disease. But for the vast majority, we don't know why amyloid piles up in a subset of people who reach the age of 85 and others do not.
Obviously, there are some ways to think about this, think about anything like this. One of them is genetic, and it's pretty obvious with the early onset, and that's the 30 to 60 group, and it's probably what Pat Summitt, I assume, has. That there's a pretty large genetic component there, right? I mean, that's pretty much.
Well, in those cases, it's what we call high penetrant autosomal dominant disease. We would say scientifically 100% genetic.
So then, I mean, the other questions, another question would be, obviously there may be a genetic factor in the kind of Alzheimer's that we're talking about here. There also might be an epigenetic factor. There probably are some epigenetic factors, right? Genes that get switched on over the course of a lifetime.
That's right.
Life kind of writes on our DNA, flips some things on that didn't used to be on. I assume that that's one of the places that the research looks, right?
Yes, absolutely. And as you were mentioning, there was just a whole slew of papers that came out in the highest profile journals about surveying those changes, those switches across all the genes in multiple diseases, not just in Alzheimer's. And so there's different switches for different genes. And I think we're at the forefront of new knowledge about that. And exactly how important that is in Alzheimer's disease, it's clear that there are switches that are being turned. How much they contribute to this issue of nerve cells dysfunctioning is something we're going to find out over the next few years, I think.
So there's a Chris Van Dyke, there's just such a thicket of questions here. I mean, how does this happen? How do we prevent it from happening? If it starts to happen, it's just in the early stages of happening. Are there therapies we can introduce at that point to slow its progression down or maybe help somebody? That's where you're coming in right now, right? One of the questions that you have is, are there drug therapies that can be introduced to help somebody if you catch it? So tell us about this study.
Mm-hmm. Well, absolutely. So, and it's very timely because really just within the past year or so, we have launched the first true prevention studies for Alzheimer's disease in our history. And the biggest one of those is something called the A4 study. the idea here is to identify people who are at very high risk of developing Alzheimer's symptoms, and then treating them in a preventive manner. So in this case, this is a study. This is going on nationwide. It's funded in part by the National Institute of Aging, and it involves looking for people who are cognitively normal between the ages of 65 and 85, but then who have evidence of early pathogenesis along the lines that Steve is describing. So amyloid plaques can be seen not only at autopsy, but also now in vivo with specialized brain imaging, specialized PET imaging. So in this case, people who are cognitively normal undergo a PET scan, and if it does show elevated amyloid, then they are candidates for the prevention trial. And the drug in question is an amyloid-lowering drug, an amyloid-lowering antibody that they're then treated with in a controlled study for over 3 years.
This is if there's some progress along these lines, either with your study or some other study, it's going to raise a really penetrating question. So, okay, you can use me as a living laboratory for a second. So my mother had an Alzheimer's diagnosis and died from it. Maybe I'm a candidate. Maybe I say, well, maybe I should be in your study. But if you have success in the study, isn't everybody going to want a PET scan? In other words, if in fact there turns out to be a therapeutic drug that can arrest the progress or slow the progress down, sometime around age 55 or so, shouldn't everybody want, whether it's practical or not, a PET scan to know whether or not they should be starting on one of these drugs?
Yeah, I think absolutely. I think so. That's an if. Mm. If we're so fortunate to have a treatment that can prevent or slow or reduce the risk of Alzheimer's disease, then I think it would become very much part of standard preventive care like colonoscopy and everything else. So that's if we're so lucky that a study like this works, and that's what we're hoping for at this point. I should add that this kind of PET scanning is clinically approved. It is clinically available, but it is extremely expensive, and really presently it's not well reimbursed by insurance at all. But I think that may change if we, in fact, have a preventive therapy, something that we could use the information to take advantage of.
We got a call here. I think it is from Doc Cayley, the science teacher I was speaking of at the beginning. The guy he's really the reason we're doing the show right now. So it's great that he's calling in, even though it's for kind of a sad reason. So you're on the air, Doc.
Hey, thank you, Colin, for the shout out. I appreciate you're not a bright middle school science student.
No, I never was. Unfortunately, you knew me at the time.
You're better than that. I should point out that. You were pretty strong at school, and thank you for your guests, their work on Alzheimer's. I'm wondering, a comment first of all, are they familiar with the work of the Alzheimer's Cure Alzheimer's Fund, which supports the research at the Alzheimer's Disease Research Foundation? And Dr. Rudy Tanzi, I would like his work, if you could comment possibly on the work that he's carrying out. I think it probably dovetails with your work. It's a slightly different approach. And also, if you could comment about the prospects for speeding research forward by their development of the Alzheimer's in a Petri dish.
All right, so this maybe we'll start with Steve Strittmatter on this one and see what he's got to say. But thanks for your call, Doc.
Very much, Colin. Best wishes to you. Thanks for putting this out there. Much appreciated.
Oh, sorry. So go ahead.
So I guess sort of the question was about the work that's being done in Boston at Rudy Tanzi's lab. He's had a long-standing interest in the genetics of Alzheimer's, been involved in describing some of those rare genes that cause the disease in early onset. But he's also been interested in genes that affect the risk, and more recently, the mechanism that your caller just mentioned, this Alzheimer's in a dish, is to take human stem cells, grow them in a dish, and then induce the Alzheimer's process by overexpressing the human disease genes in this Petri dish and then the same pathologic hallmarks that I was talking about, that is the amyloid plaques and the neurofibrillary tangles, can form in the dish. So that's been described, and this could be, we don't know yet, but this may be a way to really accelerate the research process to then understand how we can modify the formation of those plaques and their toxic effects on neurons. So it's an exciting development of a tool which may speed up research dramatically.
We're going to take a break here. Before we do that, I want to mention that Dr. Strittmatter, who you just heard speaking, is going to be honored at the Brain Ball, the first annual Brain Ball at the Hartford Science Center on March 7th. And the event's called Where Compassion Meets Science. And he will be, I think it's not like the Oscars. I think he already knows he's getting this. He'll be getting the Association's 2014 Research Award. You can get tickets to go to the Brain Ball. And this is something that, if it's something that's in your life right now, in your family, among your loved ones, you might want to think about that. It's at alz.org/CT. All right, we're back. We're talking about Alzheimer's disease. So we're fortunate to have two of the leading researchers in this with us. Christopher Van Dyke is with us, professor of psychiatry, neurology, and neurobiology at the director of the Alzheimer's Disease Research Unit at Yale University, and Stephen Strittmatter. He is professor of neurology and professor of neurobiology and director of the Memory Disorders Clinic at Yale. He's also something else, but I can't remember what it is. Really, because I literally can't remember it. I want to begin this segment a little bit, Christopher Van Dyke. There's a lot of people probably sitting out there listening to the radio saying to themselves, well, I don't want to get Alzheimer's disease. I don't even want to have it detected early enough so that I could get in Christopher Van Dyke's A4 study and have a 67% chance or whatever it is of getting this drug that maybe it's going to work. I don't want any of that. So I want to control my own destiny. I'm 35. I'm 40 years old. What can I do? And we've already said, well, we don't necessarily absolutely know what you should do or shouldn't do. You probably shouldn't play NFL football and get hit in the head a lot. But beyond that, what can you say to them?
Well, you definitely should not play NFL football and get hit in the head a lot. Yeah, and shouldn't let your kids do that, especially. But I think, this is one of the most commonly asked questions, what can we do to lower risk? And it's difficult because there are a lot of things that are suggested by certain lines of evidence. There's very little proof for any of it because we can't do controlled trials of lifestyle to prove the issue. So first of all, I would say probably the strongest evidence of all would be for things like aerobic exercise. As a general rule, we tell people that things that improve heart health also improve brain health and are likely to lower the risk for Alzheimer's disease. There's a link between Alzheimer's disease and cerebrovascular disease, stroke. There's a link with diabetes. There's some link with high blood pressure, high cholesterol. So managing all of those things is important. Aerobic exercise is probably the single biggest thing we would point to. And we're actually going to be starting this year in this national consortium that I work with called the ADCS, a controlled randomized trial of aerobic exercise. So in this case, we truly can randomize people to lifestyle, to aerobic exercise versus more couch potato lifestyle. Other things that we will tell people, there's some evidence in favor of an intellectually, cognitively demanding lifestyle. So even in older age, staying active, doing crossword puzzles, whatever things that you enjoy doing. In diet, there's suggestive evidence epidemiologically for things like fish in the diet, for fruits and vegetables, for a low-fat diet, and so forth. But one thing I would caution people, there are some programs out there that are being publicized that are really highly tyrannical of, dozens of things to do. And I would say a lot of those are not very evidence-based, certain supplements to take and so forth.
Actually, Stephen Strittmatter, as you're looking at this as a scientist, I've got a couple of different questions to ask you about this, but this is you're working on a very specific targeted aspect of this disease. But there's no way that you don't frequently pull back from that and say, wow, what kind of disease is this? How's the best way not to get it? What can I intuit about it? One thing that we know is that you don't believe that a healthy breakfast is important because you didn't have one today. But I mean, as you're listening to Christopher Van Dyke talk, does that all resonate with you, or are you more at the point of saying, well, and on the other hand, I could be telling you 10 years from now that none of that stuff does any good?
No, I think there's some consistency. What he put at the top of his list, aerobic exercise, this is something that's also true in laboratory animals who've been engineered to have something that's akin to Alzheimer's, the same kind of activity, and also exposure to novel environments are beneficial in reducing what we can detect in mouse models of Alzheimer's disease. So I think, again, not only is there clinical data, there's laboratory support for that idea, putting it at the top of the list. But there aren't too many other things.
Your work, the way I understand it, is probably more, it's pure science, but it's probably more aimed at the I've got Alzheimer's disease, what can you do for me question, right? Yeah,
our research focuses on this issue. Once there is amyloid pathology in the brain and it starts to damage neurons, how can we interrupt that process? How can we protect the neurons, even though this peptide is accumulating? Now, I would say, though, that happens really early. Most of the data now says people, and that's the whole basis of the A4 study that Chris was talking about, that people have amyloid pathology for 5, even 10 years before they develop dementia. So this is a long process. And we may in the future be able to detect those people and start them on a therapy which interrupts the biochemical, the pathologic process that happens in the brain that goes from amyloid to actually diseased brain and dementia.
Although ideally, what we'd like to do then is to break the chain before the amyloid develops, right? That would be the best thing. You want to protect the neurons after the amyloid develops.
Right.
But there must be somebody out there who's saying, well, no, let's just not get the amyloid to begin with.
Absolutely, that's true. At the present time, we don't have that therapy, number one. And number two, at least from what we know genetically right now, that would have to be something that would be safe enough to essentially give to everyone. Everyone at age 60 would have to partake of this intervention. It's very few drugs or interventions that are safe enough that you want to give them to absolutely everyone.
Yeah. No, we're not ready to put it in the drinking water yet. So let me ask both of you this question, but since he's taking a sip of something, I'll stay with you, Dr. Strittmatter. How I'm trying to phrase this correctly, but how certain are you, or what's your level of understanding about whether or not this is always the same disease, or I'm not. That's not the right way to phrase it. But obviously, it's possible that Alzheimer's is a disease that involves amyloids damaging neurons, but that if you looked at 10 patients and you could know everything about them, they might be playing out a certain way with, a really certain strong pattern in 3 of those patients, and then another strong pattern where, I don't know, maybe immune function plays a much bigger role in 4 of those patients, and then 3 more patients have some other epigenetic factor that really, in other words, is it possible that really we're talking about a disease that has multiple manifestations and therefore multiple treatments?
I think it's a very good question, and it's something that comes up in multiple areas of medicine. I guess having seen many aspects of the clinical situation, maybe Alzheimer's is more homogeneous than most diseases. So I think there's sort of less pressure to subdivide it than, say, in schizophrenia, where it may be 1,000 different diseases. That being said, though, you're absolutely right. Probably as the disease progresses, different factors become more important in different people. And to intervene as the disease goes on may require attacking different targets in order to stop the process. As you said, immune factors may be more important in one patient, whereas metabolic factors in another, vascular in another. It may be triggered early on by amyloid pathology, but then diverge into multiple steps. And the later in disease you are, the more you need to try to attack all of them, some being more important in different people.
Christopher Van Dyke, what's your take on that? 10 years from now, are we going to be talking about this in a much more multi-pronged way?
Yeah. First of all, I want to just say that Steve really did have a healthy breakfast today, because there is some evidence that coffee's actually may be beneficial for Alzheimer's disease, reducing risk. So,
coffee and a little kiwi fruit, and so that would have been, let's not let him off the hook that easily.
Oh. But be that as it may, yeah, I agree with Steve that it's more homogeneous than most diseases, certainly than, let's say, frontotemporal dementia, another common dementia. But there is a lot of heterogeneity, and I think that on a very basic level, just these early onset autosomal dominant forms are quite different genetically, age of onset, but also the way they present. The kinds of associated symptoms are really different from later onset, more common Alzheimer's disease. But even with the later onset, more common forms, there are people who carry this major genetic risk factor that we haven't talked about, ApoE, ApoE4, apolipoprotein E, epsilon 4. Within people with later onset, we've talked about this sort of common amnestic form, such as you described really with the short-term memory. But there are individuals with Alzheimer's who really begin with language variants or with what we call frontal executive function variants that can look a lot like frontal lobe dementia, and not so much with short-term memory that's relatively preserved. So I do think there is a lot of heterogeneity, and we probably will, in time, start to think of it as a constellation of diseases with a common neuropathology.
I mean, this is so unscientific, but in terms of presentation from a curbside perspective, it always did strike me. My mother was in an Alzheimer's care facility where I would visit her, constantly, and it was therefore surrounded by people with Alzheimer's disease. And I used to say, boy, they're as different as snowflakes, in terms of, some of that just has to do, obviously, with the progression of disease, where they are on that continuum. But it also did seem as just that the symptomology was different, really, from patient to patient. That's kind of what you're saying there, too. It doesn't express the same way.
That's right.
But that's different from the kind of causal questions, right? And, I just want to go back to something that you said, Dr. Strittmatter. I'm now in the area of asking profoundly ignorant questions that you shouldn't even have to be pestered with, but then you came here. So, you were we were talking a little bit more just before this about whether or not there are things, maybe different things, that kick in after the initial amyloid contamination, or whatever we want to call it. That maybe in this patient there's a bigger immune thing, and maybe in this patient there's something else. But might it also be the case, if we back further up towards the beginning of that pathway, that there are also different triggers? Like, in other words, one person eats a lot of movie popcorn, which actually has been mentioned at least in multiple articles that I read. They say the stuff that's on the movie popcorn, goes through the blood-brain barrier, and totally put me off movie popcorn. But in other words, that's a semi-facetious example. But in terms of what started this row of dominoes falling, if the end of the row of dominoes is the neurons, couldn't the differences be anywhere up that chain of dominoes that are falling? In terms of what the triggers are?
Well, I think, it would be easy to say that apolipoprotein E that Chris was just mentioning is one of the triggers, that the genetic variants there affect A beta metabolism, and therefore its ability to trigger this process which damages neurons. And so the people who carry particular alleles are clearly triggering the disease in a different way than the people who don't carry those apolipoprotein E4 alleles. So I think you're absolutely right.
And the other question I guess I have about this is, one area that are there been a lot of twin studies? I'm sure there are identical twins who one of whom winds up with this and the other one doesn't, which helps us begin to separate out the purely. You're nodding your head, so.
Oh yes. So, yeah, I mean, we do think of Alzheimer's as mostly, over half genetic, probably at least 50%. Some studies would say more 70%. But it's certainly not all genetic by any means. And one of the just most blatant examples of that is identical twin studies, where one twin may have get Alzheimer's and the other live 30 more years and not get anything at all. So it's quite clearly more than genetics. I think we don't understand the non-genetic factors very well at all. I think there's an awful lot to be learned. Certainly something very clear-cut, like prior head injury, is a known issue. Now that's something that we think can trigger Alzheimer's pathogenesis in some individuals. By the way, on that note, when we talked about tackle football, for example, increasingly I think we've come to realize that those individuals are at higher risk of Alzheimer's, but there's also something else, this thing that's being called chronic traumatic encephalopathy that's really very different from Alzheimer's disease, but every bit as bad.
We're going to take a quick break. We're going to come back with the final segment here. If you have questions, give us a call, 860-275-7266. That's 860-275-7266. You can tweet us at WNPR Colin.
Today's show was produced by Betsy Kaplan and me, Kyone Wolf. Our intern is Julia Pistel. Greg Hill tweets for us at WNPR Colin, and Katie Tolarski is our executive producer. For show pages, articles, and photos, visit our website, WNPR.org. On tomorrow's show, our annual pre-Oscar special edition of The Nose. Now, back to Colin.
And we're talking about Alzheimer's disease today. First of all, I want to remind you that on March 7th, the first annual Brain Ball will be held at the Hartford Science Center. It's called When Compassion Meets Science. Stephen Strittmatter, who's here in studio, is going to be honored at it. And you can go, if you go to the website, www.alz.org/CT. You can get information on how to get tickets to that, as long as I'm giving out this kind of information, and so I don't forget and before we run out of time. If you want to participate in clinical trials, that's what one of the things we're talking about here, you can contact the Yale Alzheimer's Disease Research Unit. You don't have to be presenting with Alzheimer's disease or anything like that to at least be considered. The phone number is 203-764-8100. I'll do that again, 203-764-8100. And it's the website is alzheimers.yale.edu. And so that's okay. I've covered everything. So one thing we haven't really talked about is the collaborative work that you're doing, the so-called FIN study. So who wants to start among the two of you down this path?
So I'll start off and tell you a little bit. Our focus in the laboratory at the bench has been in understanding how this amyloid peptide makes neurons dysfunctional. And we screened through all the genes that the mouse has, and we found a particular gene called prion protein that would capture the amyloid beta from the extracellular space and trigger bad events in the neuron. When we studied that process, we found that it went from prion protein to a protein called mGlu-R5, and then to a protein inside the neuron at the synapse called FIN, F-Y-N. And each of these three steps is the potential place to intervene with a drug. And fortunately, the third step, FIN kinase, is one that there had already been drugs developed for, because this group of molecules plays a role in cancer. But the drugs had been developed. They're safe. They didn't help cancer that much, but they have the potential to be transferred now to attack Alzheimer's disease. And so with that, and with support from the National Institutes of Health, from the companies that have really originally developed the cancer drugs, we got in touch with Chris, and we made this connection from the laboratory to the clinic, from the bench to the bedside, to take one of these cancer drugs that targets the third step in this process into the clinic. And maybe Chris will say a few words about what's going on with that trial.
So this very elegant methodology that Steve and his laboratory had worked out, we found very exciting. And So in putting together this 3-year grant that would include basic research, it would also end with an actual nationwide clinical trial for people with mild stage Alzheimer's disease. So the trial, and we've gone through a phase 1 study, looking at safety and that the drug, does get into the brain well enough. And right now we're actually just at the point of launching this national trial with this Alzheimer's Disease Cooperative Study Consortium. So the trial, we've designed a trial that I think is as interesting as the original methodology. And we're actually going to be using brain imaging as the primary outcome, not the amyloid imaging that we've talked about, but another kind of PET imaging that looks at metabolism in the brain, kind of regional neuronal activity, regional synaptic activity. So this is a way, now we can't approve drugs that benefit a PET scan, but we can very much use the PET scan at this stage to try to determine if this is, exciting enough, as we hope it will be, to go forward to a much larger trial involving, 1,000 or more people.
What we're talking about here, bluest sky, rosiest scenario, though, is the possible development or identification of a drug that would help people not get Alzheimer's who were otherwise going to get it, right? And you guys win the Nobel Prize. And,
well, I guess I would say, for mice in the preclinical models in the laboratory, using this same drug in mice that have genes that give humans early onset Alzheimer's disease, those mice are able to regain synapses and regain memory function. Mm-hmm. So the question is whether that's true for humans or not, and we won't know until this trial is complete. Then we'll have the first bit of information.
Right, and also just to clarify, now this is not in a prevention type trial. This is now in people who are very much symptomatic with mild stage dementia. But usually in our field, what we do to start with in testing in people is test them in people who have obvious symptoms, where it's easier to see effects and to see what's going on. The trials can be done in a shorter period of time. But if this particular drug worked, if it showed benefit in this kind of a trial, it could also go into much earlier stage prevention studies in future.
We've got a lot of calls coming in here, and not that much time, but let me grab a call from Sid here in Farmington. Hi, Sid. You're on the air.
Oh, that's not Farmington, but
you're
anyway,
right.
I had a question about kind of what is the natural aging process of the brain. So when you get into your 60s, all your friends are talking about, trouble remembering names and different kind of things, and what's natural to happen?
That's a Christopher Van Dyke kind of question, I think.
Well, so maybe both of us. But, I think, it's a very good question. And I think we have to emphasize, as much as we hate to admit it, that there are cognitive changes that occur as part of healthy aging. There is some slight worsening of memory, and there is some worsening of what we would call executive function as well. But it's nothing at all like the ravages of Alzheimer's disease. Now, that being said, in the very early stages of Alzheimer's, it can be very hard to tell the two apart.
Could I ask you one question, Steve Strittmatter, just going back to what you were talking about before? This is the kind of question I'm asking because there's 16 things I know about science, and this happens to be one of them. So you were talking before about sort of the sequence of things that happen and how Finn winds up to be the place where you can intervene. But the first thing you mentioned were prions, and one of the 16 things I know about prions is that's mad cow disease and Jakob Creutzfeldt and Kuru and Scrapie and all that kind of stuff, right? So are we talking about the same kind of prions?
Yeah, so there's a protein that we all have called the cellular prion protein. It can misfold and become infectious in the diseases that you talked about, Creutzfeldt-Jakob disease, mad cow disease, Kuru Scrapie. That does not happen in Alzheimer's disease, but this same protein that we all have, which can cause those infections, can also grab on to the misfolded amyloid beta peptide and trigger a degenerative process, probably the way the infectious particles. It's probably analogous to the way the infectious particles grab onto that same protein and trigger a degenerative process in those infectious diseases. But it's not the infection.
Right. We've got some calls coming in, but before that, I want to also just sort of ask you how you feel things are in terms of a societal response to this. I mean, you've got 5 million is a number that's thrown out at U.S. Alzheimer's patients. I don't know whether you know a different number, but whatever the number is, it's a lot. And so are we kind of at the moonshot stage where from President Obama on down, there's really an incredible will to fund research and pursue research that's going to stop this thing? Yeah, go ahead. You've already formulated.
Well, I think there's a growing recognition of how massive the problem is, how much almost all of us is suffering in some way or the other from it. And there's a growing commitment to make that moonshot, but we're by no means there. The way we're supporting research and discovering new ways to intervene in Alzheimer's is far beyond. It's great that things have stepped up a bit in the last few years, but if you put it in some kinds of terminology, in this country we spend something on $200 billion a year on Alzheimer's care. Mm-hmm. We spend less than 0.3% of that on research in Alzheimer's, and we don't have a therapy today. So the imbalance, very much different than every other disease the NIH supports. The support for Alzheimer's research is on the order of 1/3 what we spend on all kinds of other diseases put together.
Yeah. I agree completely, and it's very underfunded compared to things like cancer and heart disease.
And it seems as though the ratio that you just described, Stephen Strittmatter, is a recipe for breaking the healthcare system. In other words, if you've got this many patients, they need a lot of care. And we probably, over the course of that, particularly with that kind of money being spent on care, maybe get a little bit better at caring for them. And so they live 10 years instead of living 7 years or something. But if you're doing all of that and not doing the research on intervention, I mean, you're not an economist, but it seems like you're talking about a plan that will actually break the healthcare system rather than turn it around.
I don't think it requires some kind of detailed economics. The Alzheimer's problem is growing, in part because we're successful to some degrees in making progress on other diseases. The population's aging. Alzheimer's becomes a bigger problem, but we don't have drugs that change the disease course today. We need to make it that kind of moonshot effort that you were talking about, and we haven't come close to doing that.
This will be the last question, but Christopher Van Dyke, just to build on what he's saying, I'm assuming also one of the other reasons that Alzheimer's disease is becoming more and more of a problem, presenting more and more, I mean, there are a lot of different factors that would add to that, but as people live longer, they may present with a disease that we just never would have seen if they died when they were 63 or something like that.
Yeah. Well, part of it is about longevity. I mean, that was certainly the increase in the latter part of the last century. But now it's not about longevity anymore. It's really about these population bubbles like us, the baby boomers and then the generations that follow, these huge population bubbles entering the age of increased risk.
All right, we're going to have to stop there, other than I have to say, just for now, go easy on the movie popcorn with like the stuff with the fake butter. Bring your own popcorn and put turmeric on it, all right? Because that may actually be helpful. And do consider going to the Brain Ball. It sounds like a great event at the Hartford Science Center on March 7th. Thank you so much to our guest, Dr. Stephen Strittmatter, who's professor of neurology and professor of neurobiology and director of the Memory Disorders Clinic at Yale, and Christopher Van Dyke, professor of psychiatry, neurology, and neurobiology, director of the Alzheimer's Disease Research Unit at Yale University. I have a second where I can say this, and you could call if you're interested in these clinical trials, 203-764-8100. We'll put that up on our website, too, at WNPR.org. Special thanks to Betsy Kaplan, who really does understand this stuff and makes it sound like I do, too.